Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. My name is Azfa Akbar and I'm the moderator from Group 2. Today we are going to give you a short presentations about cloning. But let me introduce my team. I am Azfa Akbar as the moderator, Jesika Adilia as the first speaker, Putri Agustina as the second speaker, Jihan Rona as the third speaker, Miftakhul Jannah as the fourth speaker, and Faranida Assyifa as the fifth speaker. Our presentations is divided into several sections. First, we will explain generally about what is cloning. Secondly, we will give you the examples about how is the practice of cloning. And next, we will explain the reasons why there are pro and cons regarding cloning. And then last but not least, we will summarize our presentations. Okay then, let's start our discussions today. The first slide is what is cloning? According to etymology, the term clones comes from the Greek, that is, clones or clunes words that means twigs, tags, shoots, or hots. In a sense, cloning is duplicating step, creating an exact replica of a living creature using the creature's DNA code. Cloned organisms are called clones. Let's move to another slide with Jesika Adilia.
Well, let's move on to the next topic about why. Why? Number one, for the advancement of science. The benefits of cloning are primarily in terms of biological development, particularly embryonic reproductive and differentiation. Number two, for purposes of diagnostic and therapeutic. For example, if a couple are suspected of lowering the genetic disease of that assigment measure, they could be encouraged to have gene therapy first made with a blastomer donor. If it turns out that one of the blastomer donors contain a gene disorder that is host to that assigment measure, it is encouraged to do gene therapy in another blastomer before it is developed as a blastocyte. I'd like to move on to slide 3 to Putri.
The next goal include help infertile couple who want to have offspring, for a woman who cannot produce egg or a man who cannot produce sperm. IVF does not help. In this relationship, cloning techniques are revolutionary as infertility treatment, since sufferers do not need to produce sperm or egg. They need only a host of somatic cells from wherever they are taken, enabling them to have the gain offspring between husband or wife, to develop and propagate superior settling. If the nucleus of the donor cell is removed from a superior set, the cloned member will also possess to superior qualities. That qualities can increase even further when combined with transgenic techniques.
Well, let's move on to the next topic about when the first time cloning was found. Cloning was first made 26 years ago on July 5, 1996. Dolly the sheep was the first mammalia to be successfully cloned from a doll cell. Born in the Roslin Institute in Scotland, it has a code name 6IL3. The clone sheep named the singer and artist Dolly Parton. His cells were removed from a 6-year-old goat, and the lab culture uses microscopic needles. In the methods first used in human fertility treatment in 1970, after producing a number of normal eggs, scientists implant in replacement sheep. After 148 days, one of them gave birth to Dolly. I'd like to turn to where cloning is done. The cloning process is largely done in the laboratory. Why? Because the laboratory has many devices that support the cloning. One of the many and compulsory tools used During the cloning process is a microscope that serves to look at its cell and the membranes in the human body, animal, and plant.
I want to turn to the discussion of who did the cloning. The process of cloning is carried out by scientists to produce new animals by expelling somatic cells, such as skin cells, from the animals they want to clone. The success of cloning in animals is evident among them by John Gordon in 1962, an American scientist. Next, now I'll focus on how to do cloning. Commonly, there are some ways to make a clone. First, artificial embryo twining. The premature embryo is done by hand in separation to generate individual cells, which in turn divide and develop. Second, somatic cell nuclear transfer. Somatic cells used are cells in the body in addition to sperm and egg cells. The somatic cell nucleus is transferred to enucleate egg cells. SCNT's aim is primarily to produce embryos that will be used for research, particularly stem cell research.
Okay, now we'll focus on pros and cons about cloning. While cloning is a real of study involving various disciplines found under biomolecular and biotechnology, there are still many challenges, disagreement, or opposing sides, especially when it's come to ethic, belief, or religion and law. Problems in agriculture, such as transgenic crops for the human consumption, and then in a farm that produces a cloned animal raised motherless, could it play a natural role? Cloning of extinct species to sustain or restore ecosystem should really be needed and could replace that species at this present time. Clones sustain animals like pigs to the xenotransplantation application, cloning of cells or human organs for therapeutic purpose, and so on much requires confusing understanding and conformity to ethical, moral, and legal.
The conclusion is cloning was first made on the 5th of July 1996. Until now, Dolly the sheep was the first mammoth to be successfully cloned from adult cells. Cloning is usually done in laboratory. It has many uses and cloning have two methods. This is the end of our presentation. Thank you for your attention. It was a pleasure and honor being here today. Wassalamualaikum warahmatullahi wabarakatuh.